Welcome back to Decouple. Today I'm joined by Dr. Douglas Boreham, who is a professor and division head of medical sciences at the Northern Ontario School of Medicine and an adjunct professor of biology at McMaster University. Dr. Boreham was the 2012 Canadian delegate for the United Nations Scientific Committee on the Effects of Atomic Radiation. And interestingly, Doug also owned and operated a 50-acre vineyard and winery in one of our top wine growing regions here in Ontario. Man of many talents. Doug, welcome to the podcast.
Great to be here, Chris. Thanks.
So, Doug, today we're going to be drawing on your radiobiology expertise, some of your research on low-dose radiation in particular. We've talked about, the politics that framed the choice of LNT as a regulatory framework in previous episodes. So I think we're going to be a little more focused on the biology aspect today. But I like to sort of get a sort of personal introduction from my guests, a little bit about how they got into the field. And I understand you come from, I think, a small mining town in the north of Ontario. And if it's not difficult to talk about, I wouldn't mind chatting a bit about your father. I think he was a miner. And I think some of the occupational issues are part of what drew you into your interest in the areas you've pursued professionally. So, flesh out that introduction a little bit, tracing your journey a little bit towards how you came to become interested in the things that you've decided to focus on in your research career.
Yeah, it's interesting, Chris, how our careers evolve as we go through life. But I always started out wanting to be an airline pilot. And I was a bush pilot when I was growing up in this small town called Elliott Lake, which at the time had 9 operating uranium mines. I think at one point it had 10. So it was called the uranium capital of the world. So off I went to college thinking I would be a bush pilot. But things early on in life made it seem this whole idea of radiation was coming at me from all different directions. Probably one good example is when I was about 12 years old. I remember a person coming to our house and measuring the levels of this invisible gas called radon gas in the basements of these houses in Elliott Lake, because, of course, there's lots of uranium in the ground, and radon comes from uranium. So I remember them coming in and talking to my parents, saying we've measured the levels and they're fairly high, which means nothing to anybody. And you should probably avoid staying in the basement more than 2 hours a week. So that was the first example of advice from radiation effects that I heard. And later I questioned my parents about the fact that my bedroom was in the basement and I would spend every night 8 hours a night there, right? So they said, "Oh, don't worry about it. It's not a big deal." But so it starts this way, right? And then I play sports in Elliott Lake, and the name of our sports team I called the Elliott Lake Atoms because of uranium and stuff.
Right.
So people are always saying, "Oh, here comes Elliott Lake. They're going to glow in the dark," stuff like that. So you get used to hearing this stuff, and then finally I decided that maybe aircraft maintenance and flying wasn't going to be a good career, and I decided to go into science, and that's where I ended up going to the University of Ottawa, and then and basically for the last 35 years I've been studying environmental and medical low-dose exposures to ionizing radiation, including nuclear power.
Right. So your father was a uranium miner, is that right?
Correct.
Okay.
Yeah. So interestingly, well, I don't know if it's interesting or not, but he was a uranium miner, and of course, the mining uranium in those days, the way that it was mined clearly is not the way it's mined today, and the ventilation and the other things, including the diesel fumes that are down there and other things, really contributed to the health of the miners. And in fact, he was a uranium miner, and he did get lung cancer, and he did die of lung cancer. We had him here at McMaster, and we used a nuclear technique to find the lung cancer and then treat it. We used PET scanning and then radiotherapy.
Right.
But he basically was in a mining environment that was not healthy. And in fact, we're doing some research now on some of the miners that are still alive, because one of the things they did with the uranium miners and the gold miners was they forced them to inhale a prophylactic aluminum oxide dust called McIntyre powder.
Yeah.
So in their lunchrooms and in their change rooms, they had to inhale this black dust every day. Wow. Involuntarily, because it was apparently going to help them deal with silicosis. So it was prophylactic silicosis treatment.
Right.
And of course, today we know that inhaling large amounts of aluminum is definitely not good for you. And I think we're doing some research to look at the evidence between why, silica dust combined with aluminum dust, combined with radon gas, combined with diesel fumes, which is the class 1 carcinogen, all contributes to lung disease in miners. Of course, we don't have any of that today. And so he did die of lung cancer. But interestingly, the cancer he had was called a mesothelioma. Mm-hmm. And he was born and raised in a town called Asbestos, Quebec, which had a big open pit asbestos mine. Wow. So he had a lot of things going against him. Working in an asbestos mine before going to uranium mine, and smoking and all the other stuff that goes with mining. Wow. It does put a huge burden on your lungs. And that's why we have all this risk associated with uranium mining and radon. But if you just heard what I said, radon is only one part of the equation, yet we do all our risk assessment and analysis on exposure to radon based on uranium miners.
Right.
So that makes it very difficult for me to understand when someone says, radon could be the second leading cause of lung cancer, right, based on the uranium miner studies. Whereas we're now studying radon in some of our specialized research facilities to find out really what causes increased risk in association with exposure to all these various agents that you get as a miner.
Yeah, I mean, it sounds like mining throughout its history until we've moved into a much tougher regulatory environment for labor rights and for environmental rights as well. Was an occupational Wild West. We did a show with Jerry Grandy, who's the former CEO of Cameco, and in preparing for that, I was reviewing some CNSC documents, and they were looking at the feasibility of studying the health impacts of radon. But they basically discovered in preparing for the study that the radon dose to the miners in a contemporary mining environment, a uranium mine, was lower than the dose they'd receive at home from radon gas in their houses. So, they said this study is just not feasible to do. And I'm sure there was probably objections to that from certain people who said, this is a, regulatory capture, example or something, but the radon doses are just so low now. The occupational standards have gone so high that I think mining has become a very different thing, at least in the developed world, in a highly regulated environment compared to, like I said, those Wild West days that your father was, and the idea of being forced to inhale McIntyre powder that's wild.
Yeah, it's quite a story. And if your audience is interested in looking up, there's some work being done now in Ontario, and it's being led by a lady named Janice Martel. Right. And she's the one that's been the driving force behind it. And there's quite a bit of information on the internet about this McIntyre powder. It's interesting as a foundation that was created and made money by insisting that all the miners in uranium and gold were to inhale this stuff before a shift. And it got even over to Australia, where Australian miners were getting the same thing. So it's quite a story. And there's a story. It's a very scary story.
Yeah, definitely. I mean, I could nerd out about uranium mining for a long time. I think, there's so much I want to draw out of this interview. So maybe we'll move on here. But I mean, I guess in approaching this topic, it probably makes sense to just get some basic radiobiology out of the way. So we're speaking kind of a common language. Again, my audience tends to be pretty literate on this topic, but, a couple of things I just wanted to go into. I think a good definition of LNT with some of its peculiar assumptions would be useful to hear from you. So, I mean, we've heard about things like, cumulative dose, et cetera, but can you just kind of go into what you believe are the major flaws in the LNT assumptions?
Yeah, so I have a lot of experience working with the LNT. So the linear non-threshold simply means, as you know, that any dose of radiation is going to have a risk to it associated with it. And this has all been derived from exposures to high doses where you can see effects. And we know that like high doses of radiation do have detrimental effects, whether they're immediate, like skin reddening or a tissue necrosis, or long-term effects where you get things like the development of cancer. So this data has been derived from really the only human experiment that was done, and that's the dropping of the atomic bombs on human populations, right? So, and it was discovered over the years that people that were exposed to high doses of radiation from the atomic bomb were developing increased risks of cancer. And more recently, there's been some other associations with perhaps cardiovascular disease in the high dose range. I guess the question becomes, where does the high dose range turn to a low dose? And when does a low dose maybe not follow the linear non-threshold hypothesis? In other words, the threshold being at some point it's not harmful to you.
Right.
And I believe now that we have substantial scientific data, including modern molecular tools to study the biological responses of cells to these types of stresses, including the radiation. But back to the sort of experiment with the lifetime study of the atomic bomb survivors. And interestingly, a lot of atomic bomb survivors were young children, especially in Hiroshima, because during that time period, there was a lot of bombings going on that involved fire bombs and stuff. So a lot of the Japanese cities were being burned, and you'd imagine the construction in those days was a lot of wood and paper and other things. So very, very dangerous. And I believe, some 80,000 people died in Tokyo because of fire bombs. Mm-hmm. So during the Hiroshima incident, apparently there were a lot of children that had to come into the city because they were brought from the country where they were safe into the city to help clear out firewalls and fire breaks in case, another fire bomb attack came. But in fact, what happened was an atomic bomb. So a lot of young children got exposed. So we have a lot of them still alive today. So we have an opportunity to study a very long time frame for these cancer risks. And the issue is that at a certain point, it's very difficult, if not impossible, to show that there is an effect at the low dose range. So you have to rely on mathematical modeling. And so what people have done in the past is they've looked at this model and said, okay, we know that if you get exposed to a dose that we call, say, 1 gray or 1 sievert, it's a fairly high dose, that the risk of getting cancer from that over your lifetime would increase about 4 to 5% above what we already have today. And I believe we're approaching 50% in the population, at least, will get cancer over their lifetime because we're living a lot longer,
right,
staying healthier longer, and cancer. So the risk goes roughly from 50% to maybe 52% or 54% if you've been exposed to a high dose of radiation.
And who gets a gray? I mean, this is
who
not,
gets a gray.
Right?
No one gets a gray. Well, some Chernobyl workers got a gray, or more. You start seeing effects that we call acute radiation syndrome, where when a human gets exposed to doses above a gray, then you start seeing immediate effects, which could be simply nausea and vomiting, dizziness. Basically, what happens is, depending upon the dose, but at a gray, you have a risk of your immune system being suppressed or depressed. Right. So your white blood cells are depleted, and your risk of dying of an infection goes up. So that's kind of what we call the hemopoietic syndrome. So who gets a gray? Well, some people that are unfortunate and get caught into a situation where there's an accident, where they're nearby. So, for example, Chernobyl had nearly 30 people got exposed to doses that caused that gray and higher effect. Right. There's numerous little incidents that have happened over the years where people working in, say, sterilization facilities have bypassed a lot of safety systems and managed to get themselves, 5 gray or 10 gray and died from it. Wow. There's been situations.
That's in medical applications?
Well, no,
or sterilization as well.
This is sterilizing food, I think, in I don't remember the country, but it's over in Europe.
Okay.
And somehow something got jammed up in the system. In other words, it's on a chain system where boxes of things are brought in and out of a room that has high levels of cobalt-60 gamma radiation in there, which sterilizes stuff. But somehow it got jammed, and this individual decided that they would jump across a barrier that was to prevent people from going in there, turn off all the safety systems, go in and unjam the chain. And by doing that, expose themselves to a lethal dose of radiation. So stuff like that happens occasionally. So we know that there are people that get exposed to these high doses.
But I guess really like in human history, that would probably be if you exclude radiation oncology patients who get many, many, many grays in fractionated doses, excluding that population, nuclear accidents, sterilization accidents, atomic weapons testing, or the Lucky Dragon incident, we're probably talking like 1,000 people, maybe less.
Oh, maybe less. Yeah. I mean, these individual events, we're talking less than 100.
Right.
So, but when you start grouping into that atomic bomb testing and other large populations that may get exposed, they would not get 1 gray of radiation. That's certain. So they would not have those types of acute radiation syndromes, things we're talking about at 1 gray.
Right.
But, what happens is we look at this and we say, okay, sure, we have an increased risk in the populations that got 1 gray and above. There is no increased risk typically above 4 gray because people died after 4 gray. It was too high a dose. So they were too close to the hypocenter of the bomb, and so they died. Typically, a lot of them would have died because of the thermal blast, not the radiation. So these are very immediate effects.
Yeah.
And the other thing that people don't take into account is the fallout. So there was this thing called black rain. So for days after, there was radioactive fallout actually coming onto people that weren't actually there when the bomb went off, but came in to help people. And they got exposed to this black rain that then caused them to have acute radiation syndrome, but there's no way to know what dose they got.
Right.
And this is the other problem with the LNT is it's based on the dose that people got when they were exposed to the bomb. And the dose that they got is based on the distance you're away. So as you know, the further you get away, the less effect you're going to have. And far enough away, you get a low dose, and then you'd have to start figuring out. So initially, to figure out what dose people got, it was, where were you when the bomb went off?
Right.
And that was fine until the government or the society started saying, well, if you're near atomic bombs, then you're probably a mutant, and we don't want to marry you, and we don't want to have kids with you, and we don't do that.
Right.
So almost within a short period of time, people all of a sudden became further away from the hypocenter. So they weren't actually exposed to the dose they said they were, which caused a bit of a problem when you're trying to estimate risk on the dose you got, but now you're not really telling the facts on where you were.
Right.
Then another change happened where the government believed that they could compensate people. So you're actually getting some monetary value from being close. And all of a sudden people
got closer
to the bomb.
Right.
So working out the dosimetry was very difficult.
Right.
And the other thing that happened, of course, was the thermal dose was significant. There was no accounting for the fallout doses that people got. It was basically based on the distance you're away. So after 4 gray, we have no long-term studies we can work on. Roughly below 1 gray, there's not enough statistical power. There's statistical power still at 1 gray to say it's 3 or 4%. But as you go down, you can imagine that the number of people you need to be exposed in that dose range has to be significantly higher to get statistical power. And at some point, the lines, the error bars on what we call the dose and the low dose range overlap the zero axis, the x-axis, right? So we have no way of knowing based on this hypothetical model. And so that's why when we look at the LNT model, you can extrapolate, and people do, to low dose ranges. But I think you have to look at, when it comes to LNT, what are the, what hypotheses are you testing here? Yeah. So with LNT, dose is proportional to risk. Dose adds up. So, a little bit now, a little bit later, and a little bit later after that adds up. So dose is additive.
And that's in direct contradiction to, the practice of fractionation of doses in radiation oncology, where multiple grays are delivered to focused tissue, and you break it up so the tissue has a chance to recover. Like if you applied LNT to radiation oncology treatments, all those patients should be dead, basically, right? So there's like a direct contradiction there. Am I right on that?
Yes. Yeah. So
like generally in science,
argue that.
Sorry, go ahead. I mean, generally in science, like you have to prove a null hypothesis, and if there's conflicting data, like you kind of have to throw out the theory.
So the null hypothesis in this is that the risk is the same at low doses and that is at high doses. And in fact, if we look at the scientific data, we can show that it doesn't follow a linear line for almost every biological endpoint we look at. So you kind of have to reject the null hypothesis and say there is a difference in the biological response at high doses compared to low doses. Right. And that's where the science starts coming in. And as we get more and more into high-powered molecular genetics and molecular biology, we're starting to understand the pathways. And now we can look at all pathways with all the omics that we can do. And we're really getting a good understanding that cellular responses in the low dose range are not the same as cellular responses at the high dose range. Consequently, then risk would not be proportional to dose in the low dose range. Right. And so if you look at the models that people have proposed, much of the research we do shows what we call a J-shaped curve, which means there is a threshold for risk. And at some doses, the threshold for risk actually goes down. So the risk is less than 0 dose. So that means it's probably not bad. It could be beneficial. Mm-hmm. So by taking all these things into account, we can start looking at the LNT. The idea of using high dose to extrapolate risk at low doses is probably a good thing. Right. And the LNT also assumes that we're all the same, right? So dose is additive, risk is proportional to dose. We're all the same. So our response to radiation is going to be the same, right? Because you're extrapolating and you're saying this number of 10 millisieverts gives you a risk of this. well, we're all not the same. In fact, we're all different. And there are many things that make us different, including genetics, but also environment. And we know things that we do environmentally, whether we exercise, whether we eat, what we eat, the types of things we do like sauna or take jacuzzis and stuff like that changes the biological response of our cells at low doses. So we're not all the same. We're not all the same genetically. And so all these assumptions in the LNT are not supported by the data.
Right.
So at some point you have to sort of reject the LNT hypothesis. And so obviously you can tell the way I've been talking, I've never really even accepted it, even in the beginning back 30 years ago, because we had data that says, well, it's just, biology doesn't work in a linear way.
Right.
And so we need to understand what is going on at the low doses that makes things happen that you can't predict from high doses.
So, I mean, biology has grown in leaps and bounds, particularly molecular biology. One of the things I heard was that the initial conception of DNA was that it was incredibly fragile. This, I think probably because of the methods that were used to discover its structure. What I'd heard is that early geneticists, and they weren't aware of repair mechanisms, and so there was this idea that here's this strand of genetic information, and if it gets disrupted, it's not going to recover. I don't think we had the same understandings of, programmed cell death, et cetera. So is that true? Has that really changed our understanding? Because I think there's a real lack of appreciation for repair mechanisms, and we sort of...
Yeah.
Yeah.
Well, there's two sort of things, and it sort of goes back to, if you think about when the notion of LNT came out, it had to do with research done on fruit flies and mutation rates, right? And it was reported that, in fact, these mutation rates in fruit flies were linear in proportion to dose as you went down in dose. That's been very controversial. There's lots of people you can talk to about that research and whether or not that should have been adopted initially by the Americans as part of risk associated with exposure to chemicals and radiation. But that being said, we had a lot of various endpoints that we looked at that can show us from a molecular perspective that things are really a lot different. Now, you just asked me the question about, I kind of drifted there a little bit.
Yeah, no, this idea that, when DNA was first discovered, that it must be this very fragile molecule and a lack of appreciation for repair mechanisms.
So it's interesting because DNA is very, it should be very fragile. When you think about it, each one of your cells has about a meter's worth of DNA in a cell, in a single cell, right? Mm-hmm. So a meter's worth of DNA is a lot of DNA. It packed into a tiny, tiny space like the nucleus of a cell. And that DNA has got to be actively repaired. It's actually metabolizing, it's transcribing genes. All sorts of things go on that DNA. It's being replicated. It's being hammered by all sorts of environmental agents like natural free radicals and other environmental toxins. Yet it seems to maintain its integrity. And so that 1 meter of DNA in each cell, if you add all the cells up in your body, 100 trillion cells, you can stretch your DNA from the earth to the sun and back hundreds of times. That's how much DNA you have packed in the little body of yours. And now you're saying, now we have to have some way to keep the integrity of that DNA from falling apart, because it really only takes a small change in a little bit of DNA to cause one cell to go rogue, right? It's not going to pay attention to the cell signals, which I find, I don't really understand how we can actually be alive on this planet with all this insult, with all this DNA that needs to be maintained. And so the way we look at it typically is remember that even before 1956, we had 48 chromosomes as humans, you know that, right? And all of a sudden someone discovered by mistake that in fact they used the wrong solution and they made a hypotonic solution where the cells blew apart and we could actually count them and there's only 46. Wow. So people accepted that big change in genetics almost overnight because we didn't have 48 chromosomes, we actually had 46.
Right.
And from that point on, we've been learning more and more about DNA and how it pairs itself. But in a simple sort of, scenario, you could think about DNA as being intact until it gets damaged, say, for example, like by a chemical or by radiation. So basically the cell, three things can pretty well happen. The cell can repair itself. And there are a number of DNA repair systems that we use. I mean, ultraviolet light is a classic example. You don't put sunscreen on, you go out in the sun. Ultraviolet light's going to damage your DNA by cross-linking it. If you don't have the repair mechanisms to repair that cross-link from the sun, then the cell dies. If enough cells die, then you get like this reddening and the skin falls off you. And that's kind of, everyone's experienced that, I'm sure, at least once in their lifetime. Yeah. There's a classic example of too much DNA damage and not enough DNA repair capacity to fix those crosslinks. There are genetic syndromes where if you have a defect in the one gene or the pathway that fixes crosslinks from sunlight, then your risk of getting skin cancer goes to 100%. So you've got to avoid exposure to ultraviolet light. And this gene is called xeroderma pigmentosum. There are people that have that genetic defect that can't get exposed to sunlight, or they will get skin cancer. The challenge that we have is we don't, and these people have a knockout in both copies of that gene. So that they've inherited a bad copy from their mother and a bad copy from their father. Right. And therefore, they are very sensitive to ultraviolet light or DNA crosslinks because of one DNA repair pathway. Yeah. There are other genes that are associated with ionizing radiation risk. And that means that you can't do a certain repair of a type of damage caused by ionizing radiation, which is different than ultraviolet light. Yeah. It doesn't cause crosslinks. It actually breaks the DNA. It can break a single strand of DNA. Or
is that from a direct hit, or that's from oxidizing
So it depends on the quality of radiation. If you're talking about, say, something you would do in the hospital, like an X-ray, we call that a type of quality of radiation is low energy transfer radiation. So that's low LET. And that type of radiation from your X-ray machine interacts with all the molecules in your cell. And because your cell is mostly water, the water goes through radiolysis and breaks into these reactive oxygen species. Basically, the water breaks into a hydroxyl radical, a proton, and an aqueous electron. So these reactive oxygen species that are created by your X-ray of your low LET radiation then are reactive chemically and damage your DNA. And basically, that's what happens most of the time when we're talking about medical diagnostic radiation. Is the LET of it is such that most of the damage caused your DNA is what we call indirect. So it's not the photons themselves that are causing the damage. It is more like the chemicals that are produced around your DNA, the reactive oxygen species that does the damage to your DNA. So indirect damage.
Gotcha.
So we have very efficient systems to deal with that, scavenging those reactive oxygen species. We have proteins that can suck them up. We have DNA repair systems that can go and fix the double-stranded DNA or the single-stranded DNA breaks caused by these free radicals. And all these are genetically programmed in our cells. Mm-hmm. So consequently, if it was a perfect world, we would have all the proper DNA repair mechanisms you need for the various types of DNA damage caused by ionizing radiation or ultraviolet light or chemicals. And then we would probably live for a long time because our DNA would stay intact. The other super powerful systems we have is that at some point our cells have recognition systems and checkpoint systems where the cell itself, cells that are dividing are typically more sensitive to radiation because they're replicating DNA and they're trying to get one cell to another with having no breaks or any damage. Right. And so dividing cells are typically more susceptible to the effects of ionizing radiation. So when we show that these dividing cells are more susceptible, there are checkpoints that a cell has that says, okay, wait a second here. I'm about to go and divide, but I'm not really ready to divide. So these are called cell cycle checkpoints. And those checkpoints are very important for our cells to take a break and repair themselves before they divide and make a mistake. Mm-hmm. So if they divide and make a mistake, that mistake could be transferred to one cell or the other, and then that cell can be the one that becomes the ultimate cancer in your body. But the checkpoints help slow down things and allow DNA repair to happen.
You'd mentioned this low energy transfer. Presumably there's a high energy transfer, and that's a different mechanism for DNA damage?
Yes. Yeah. Well, let's say, for example, we'll take an example again in medicine where you want to treat a tumor with a very high LET radiation, which is what we call, say, alpha particles. So there are medical isotopes that are being produced today that give off very high energy alpha particles that interestingly don't go very far. Mm-hmm. And I think the analogy is if you look at the low LET radiation, it can penetrate a lot of things, which is why an X-ray works. It goes through your body. It's shielded, the bone shields a little more than the soft tissue, and you can discern the difference between structure and soft tissue. But higher LET radiation doesn't have that penetrating power. Yeah. Very short distance, without a very short track length. So the high LET stuff, so the alpha particles that you might want to treat a tumor with, can only travel maybe the distance of a nucleus of a cell or two cells. Right. And they're like a cannonball going through a cell. They just They just plow a trail through the cell and they directly damage the DNA. They don't make many free radicals, but they plow right through the cell's DNA. And it's like firing a cannonball through the nucleus. It just blows all the DNA apart, which is why these things, if you get them into your cells, they're very damaging. And this is what sort of we use for using various types of isotopes to treat cancers. The high LET stuff, brachytherapy works the same way, where you have a very high energy, low track distance. So the idea is you want to keep all the energy from the ionizing radiation very close in the tumor or to the tumor and spare the surrounding tissue, right? Because as you know, as a physician, we can cure all cancers. The challenge is to keep the patient alive. And in other words, keep their normal tissue functioning. Right? It's if you talk to my colleagues that are radiation oncologists, they say, well, our challenge is always to keep the normal tissue alive so the patient stays healthy and has a reasonable quality of life after going through the therapy.
There's a famous expression in medicine in general that, the operation was a great success, but unfortunately the patient didn't make it.
Exactly.
Okay, so there's this high energy transfer, and that's, you said, from alpha particles, which is, very local effect, blocked by a piece of paper, for instance, but if taken internally, can be useful in cancer therapeutics. There's this idea that's touted around by a lot of anti-nuclear folks about hot particles. So we have in Toronto, uranium processing facility. Like, we used to be obviously an industrialized city, so there's still some fabrication factories that take mined uranium and pellet them and process them, et cetera. And there were people going around our local neighborhoods, to school playgrounds and saying, listen, if you inhale a single particle of this uranium, it will deposit in your lungs, and because it's going to be a high energy transfer, as you mentioned, it's going to, one single atom could be enough to cause cancer in local tissues. I know that when we talked about grays before, sieverts is more of the biological effect. And that takes into account, the route of exposure and the sensitivity of the tissue exposed. Can you tell us a little bit about that hot particle theory? Because it sounds pretty scary.
Well, it does sound scary. And in fact, if you have, it depends upon the size of your hot particle, right? That's a pretty loose term. If you're saying a single atom, then I don't call that a hot particle. I call that a single atom that's going to decay. It's going to give off a very low dose of radiation. One thing about alpha particles is because they cause so much damage, they cause a lot of death. And dead cells can't become cancer cells.
Right.
So by having a very high level of high energy radiation hitting a cell, there's a good chance it's going to die. That's another one of the mechanisms we have that protect us from this kind of stuff, is that we have mechanisms that tell us or tell ourselves to commit suicide or program cell death, right? So that's the other very powerful pathway, other than DNA repair. If the repair can't manage it, say from the complexity of the damage caused by a high LET alpha particle, then we have mechanisms in our cells that can simply say this cell is irreparable, and it has to self-destruct. Because, like I said, again, a dead cell cannot become a cancer cell. Mm-hmm. So we have powerful mechanisms that will cause cells to commit suicide and eliminate themselves from the population so that they do not ultimately become a cancer. So cells that are these hot particle theories, unfortunately, we are full of radioactivity, right? We have lots of uranium in our body. We have radium in our body, radium in our bones, giving off alpha particles all the time. We're looking at thousands of decays of radioactivity in our body every minute, every second, actually, 7,000 decays roughly a minute from all the natural things we've eaten and we've inhaled or drank, or like you said before, if you have alpha particles on the outside of your body, even the layer of dead skin cells can stop an alpha particle from hitting a living cell. When it gets into your body, then it gets incorporated into your various tissues. Mm-hmm. And if you're talking about a radioactive material called tritium, which people are really concerned about when it comes to nuclear power stations, especially the Canadian design ones, tritium is a naturally produced radioisotope. It's in our water naturally. It's caused by cosmic radiation interacting with water molecules or hydrogen in the air, and it becomes tritium. So your glass of water you're drinking, or all the water we have has somewhere around 5 decays per second of tritium in it. So you're drinking that every day. Mm-hmm. And then we have other isotopes, like I said before, we're eating lead and polonium and radium and all these things we can inhale and eat. And it basically, we become radioactive. The two things that are sort of naturally occurring that are in our body are the potassium 40 which, as you know, potassium is important for nutrition, and that's why you want to eat your bananas. But part of the potassium in those bananas is the radioactive form called potassium 40. And from a challenge for us in the radiobiology world is we're trying to figure out what happens to cells when absolutely no radiation is around them, like none. And our challenge is to get rid of potassium 40 because it's everywhere. And you can't get rid of it, right? No. The only thing you can do is you can find the stable isotope potassium 39 and try and displace the radioactive form of it, which is what we're challenged with right now. But so potassium 40 is a big one that's in our body, and you're giving off thousands of decays of that.
And that's very, very high energy beta decay compared to tritium, for instance, right? Because that's right. What you hear a lot. Well, tritium is, it acts like water, so it's going to go throughout the entire body. It might end up right close to DNA, but potassium is the predominant positively charged ion within cells themselves, and gives off, I think the decay is like 80 times more powerful than tritium. I think like the key takeaway for me is that the world is naturally radioactive. Our body's naturally radioactive. If we didn't have the repair mechanisms in place, we'd be up shit creek. And
wouldn't be
one thing that was really interesting that I heard you say, just the other fact I heard a little while ago was that we have 10,000 single strand DNA breaks every day. And I think, those are much less of a problem because they can be repaired quite easily. And then I think it was
probably every hour.
Yeah. So, I mean, it's very, very dynamic. Like our DNA is constantly being damaged, constantly being repaired. In terms of that low energy transfer radiation, how does that compare to, the production of free radicals as a result of our aerobic metabolism? Like what's the ratio, I guess, of damage from radiation versus just our day-to-day metabolism? Because you had this example I heard on another podcast, actually, the South Bruce Willing to Listen podcast hosted by Sheila Whittock. And you'd mentioned, a patient that's just waiting to get a CT scan, in the waiting room would get more DNA damage just from being alive and breathing oxygen than from that CT scan. That there'd be more DNA damage. Can you run me through that again? Because I was, as a medical doctor, orders CTs all the time. Yeah.
You're a good learner, Chris, because it's exactly what I said. And it has to come down with oxidative metabolism. And the thing about it is, we have mitochondria in our cells, which is the little energy factories that keep us alive and give us energy and produce energy. And so producing energy, you're running a little engine inside our cells called mitochondria. And those engines, they need water and they need electron transport, and they need a number of other processes that create free radicals. That's the byproduct of breathing air, is that you're going to create what we call these reactive oxygen species. So oxygen we breathe in is used to make the energy. But the byproduct of the oxygen is a reactive oxygen species, which then is a DNA damaging agent, just like radiation. So in fact, breathing air causes DNA changes in our DNA at a pretty significant rate. And the more air you breathe and the higher amount of oxygen you have, as you know, if you're giving someone oxygen that's above 20%, which you have to do in some circumstances in your emergency room or in the clinic, right? It causes damage to the lung tissue because of the free radicals you're producing with high levels of oxygen. Yeah. And everyone knows how hydrogen peroxide works, right? If you have a cut, you go to the pharmacy and you put hydrogen peroxide on it, it starts fizzing and burning like heck. Yeah. Well, that's another form of reactive oxygen species that's created inside our cells naturally from metabolism or by ionizing radiation. So again, we're just talking chemistry here. And so the ionizing radiation producing hydroxyl radicals or superoxide radicals, or the same thing produced naturally in our cells, is causing this disruption in our DNA and our biomolecules. So we have all these processes that we've evolved to protect us from that. And I think the perfect example is if you're going to get a CT scan, the dose of radiation you get from that CT scan is going to cause some DNA damage to your cells. We would say a CT scan would roughly put 4 photons through each cell in your body. Wow. So that's roughly what you get. That would cause some small amount of DNA damage. But over the hour, those 4 tracks of radiation would be equivalent to, say, 400 tracks of radiation by breathing oxygen. So about 100 times more things are going on just by breathing the oxygen compared to the CT scan. Wow. So when people are worried about going and get the CT scan because there is this increased risk, they're sort of they're not aware of the fact that the DNA that's going to get irradiated is already being naturally irradiated by reactive oxygen species from endogenous free radical processes. And so I like to use that analogy because it's like when I go to the dentist and they throw a big lead apron on me and I'm going, the dose of the radiation coming off that dental x-ray is not even enough for me to even, worry about it. You wouldn't even worry about it. It's so tiny. It's like we're called microsieverts. And compared to the amount of stuff I put in my mouth and fry the inside of my cheeks with stuff I eat that's super, toxic from hot sauces and other things like that, I'm not too worried about the dental x-ray. So it's a very interesting situation when you talk to the free radical biologists, which is where basically the theory of aging has come from, free radical biology, which was adopted through radiation biology because the processes are very similar. I mean, we can artificially age things with high doses of radiation. And, the reason you get gray hair is because of these reactive oxygen species in your hair follicles, causing the pigment to be bleached away. It's like putting peroxide on your hair. So you turn gray. So these are natural processes, right?
Yeah.
So, you're right. And in fact, the whole amount of endogenous free radicals causing DNA damage to our cells is probably part of the reason why some of us are more susceptible than others of developing disease later on in life. Because like I said before, the LNT assumes we're all the same, but as you know, we're not all the same. And if you're missing that one DNA repair pathway that you need to, say, fix a single strand break or fix a DNA crosslink, then your cancer risk gets significantly higher. And again, I said earlier that on average, I don't like putting everyone into an average box, but I will show a picture of an average human female and an average human male, which you can find on the internet. Just look up the average human face. And that's a combination of 1,000 different people. And that's what they look like, but they're not real, right? They were never born and they're never going to die, but they look like a real person, but they're an average person. But we're not all average. And so some of us may have zero risk from an exposure to something, and others may have 100% risk. But on average, we get this average risk. So a lot of people argue that average numbers don't apply to you or me. Mm-hmm. They apply to the average person who doesn't really exist.
Right.
So, and like for example, early on in my career, we were looking at things that modify the risk of, say, death by radiation. And this is sort of the way we used to do experiments in the old days, and not me, because it's very highly regulated now, and you just don't irradiate things to see how they die anymore, because it's, unless of course they're a yeast or a bacteria. But there are studies that show if you take a large population of mice and expose them to 9 gray, for example, which is lethal, to say 200 mice get 9 gray of radiation, and within 15 days, every mouse is going to be dead. Right. Because it's just too much radiation. I mean, that's 9,000 tracks of radiation through every cell in that mouse's body. So they're going to die of very likely the hemopoietic syndrome or the GI syndrome. It means their gastrointestinal system has fallen apart or their immune system has failed. Most likely with 9 gray in mice, it's their immune system. So it takes about 15 days for them to die of an infection or something. Right. But a group of scientists in Japan took same genetically identical mice from isogenic, they're called. And they, before they gave them this lethal dose of radiation, so the 9 gray of radiation, the day before, they basically put them into a sauna and increased their core temperature by 1 degree Celsius for 1 hour. Right. And what that did in every cell in their body is it caused their cells to go into what we call heat shock response or stress response. And the cells then turn on a genetic mechanism that's highly conserved in everything we've ever studied that's alive, that simply says, there's a heat stress coming, prepare for death by heat, and forget about all your other functions. So the cells stop dividing, they start synthesizing these heat shock proteins and other chaperone proteins to keep DNA from falling apart, including DNA repair proteins, getting ready for the big shock that's coming. Right. Well, a 1 degree temperature shift, as you know, is a mild fever in a human, right? Right. And that's all this is. It's 1 degree elevated temperature in a mouse for 1 hour, 24 hours before getting 9 gray of radiation. Yeah. The experiments showed that 3 months later, every mouse was still alive after 9 gray radiation from a 1 degree, 1 hour hyperthermia treatment. So that just tells you right there, that cells have the ability to adapt, and they have the ability to respond to insults like a serious amount of radiation, and they survive. Wow. Whereas they wouldn't survive without the environmental stress of a bit of heat. So, and I can go on and on and on about all the different things we can do to manipulate molecular pathways that will increase or decrease the risk of an exposure from radiation. But don't get me wrong now, radiation, if I really wanted to study carcinogens, radiation is a poor carcinogen. It takes a whole lot of it and a whole lot of time to get a small increase in cancer risk.
Yeah.
We use chemicals in the lab that are significantly more potent at causing cancer, like alkylating agents, DNA cross-linking agents. Typically, all the things we use to treat cancer can also be used to cause cancer, right? And so we study how those processes can be manipulated and how they, those molecular mechanisms can be modulated. But there's no reason to believe that your response to radiation today is going to be the same tomorrow or the next day. It depends on what's going on, right?
Right. No, I mean, it's very interesting comparing the oxidative stress versus radiation-induced damage. In medicine, our clinical practice has changed pretty dramatically in the last probably 5 to 10 years. We used to just think, hey, oxygen's good. Let's just blast everyone with oxygen who's sick. If you have a heart attack and your oxygen levels are normal, we'd still put oxygen on you because we'd think, well, we can get more oxygen to those cells that are under stress because the artery's blocked. And we found over and over again recently that it actually increases mortality. Newborn babies, they naturally don't have, they're switching over their circulation. So their oxygen levels are low for a while. And now, it's interesting how that's changed just in my career so dramatically. And it's, because of actual increase in death as a result of hyperoxia. So it's very, very compelling.
You mentioned that there is some, and you may want to have someone on your podcast in the future that talks about adaptive medicine. I go to a lot of conferences. They're called low dose adaptive medicine conferences, and all sorts of things like you talked about. When you have a heart attack, there's ways that even after the heart attack, you can reduce the cardiac injury by manipulating other parts of your body, right? And one example is some researchers just put a blood pressure cuff on one arm or one leg of a test animal, and cause it to go hypoxic after heart attack. Then releasing that into the blood system reduces the damage to the heart later on. Wow. So it's really, really cool medicine. I think it's be worthwhile to bring that in and compare it to adapting with things like low doses of radiation. Yeah. And I think where we're going to see in the next 10 or so years, and it's even happened a little bit during COVID, is that people are starting to understand that using low doses of radiation can modulate your immune system, modulate the way our cytokines work, modulate the cytokine storms you get by infection. People have used ionizing radiation in history to prevent infections, or to get rid of infections, or to prevent them before surgery. This is all before antibiotics were invented, right? A typical procedure might have been, you get a whole body X-ray before you go into surgery because your risk of getting infection is going to go down because it stimulates your immune system. Wow. And we're doing studies right now where we're looking at how low doses of radiation can stimulate immunity in a way that it can be a cancer fighting agent.
And I mean, I think a really interesting thing you were telling me about is you have a lab is it 2 kilometers underground? It's lead lined. You're feeding I'm not sure if it's plants or animals, potassium 39 only, and you're trying to get to a zero radiation environment. And I think you mentioned there is some previous research that's analogous to this that showed that the plants didn't actually thrive or actually suffered because there was no radiation, no stimulation maybe to these repair mechanisms. It's fascinating area, but I think that's just a cool story. So just tell us about that and what it's like.
So back many years ago, it was people were testing the hypothesis that the natural radiation that we find around us is essential for life processes, like all the things by stimulating all these DNA repair mechanisms or these apoptosis mechanisms. This is stimulated by exposing ourselves to these mild stresses every day, and natural background radiation being one of them. And so people were testing the idea of taking away natural background radiation. And for example, you could take yeast cultures, which are a rapidly dividing eukaryotic cell. They double every 2 hours roughly. And you can study their growth rate and their DNA repair under normal sort of natural background levels you would have in a lab, or you can shield them from cosmic radiation and other types of radiation in lead caskets that aren't radioactive and things like that. And you can watch their DNA doubling time go down. You can watch their DNA repair capacity just get worse. So the number of mutations goes up, the number of double strand breaks goes up. So you can study that very effectively. And then what they did was they said, okay, well, we're going to reintroduce, even though they're shielded now from natural background radiation, we're going to reintroduce them to an external source of radiation equivalent to the doses of background. And under those conditions, the cells responded and went back to normal growth rates and normal DNA repair. It was done in different organisms, small sort of little amoeba type thing was looked at by taking away potassium 40, which we had mentioned earlier is one of the naturally occurring radioisotopes that contributes to a lot of radiation from natural sources in our bodies. And by taking that away, the same thing happened. The growth rates disappeared. Their ability to replicate and divide went down. And again, reintroducing it as an external source recovered the animals or the organisms, and they were fine. So our project and our experiment is down in the SNO lab, which is the Severing Neutrino Observatory, which is significantly shielded from 50 million times less cosmic radiation than you're getting right now, sitting here talking to me from cosmic rays or cosmic space radiation. Yeah. So we eliminate that by 2 kilometers of solid norite rock. And then we eliminate the radon gas that's in a mine, it's in all mines, by simply pumping in old gas, so old air. We'll get air tanks that we let sit around for a couple of months and all the radon decays away. So now there's no radon in the air. So now you have clean radon-free air. And so we take away the radon, we take away the external gamma from the walls, we take away all the cosmic radiation. The last challenge is take away the potassium 40 and the carbon 14, but carbon 14 doesn't account for very much of our natural stuff. Right. It's good for carbon dating things, right? I mean, as you know, we eat carbon 12 and carbon 14 and carbon 13, and it's always in an equilibrium, in a balance. And as soon as we stop eating, the carbon 14 starts disappearing. So you know what happens when you stop eating, right? You die. Yeah. So people can look at bones, say, that are 5,000 years old and say, these bones stopped eating about 4,000 years ago or 5,000 years ago based on the carbon 14, carbon 12 ratio. Right. So it's kind of a cool tool, but it's still in our bodies, right? We're talking carbon 14 right now. So in this lab, we are developing experiments that are going to look at cancer risk in human cells, the ability of eukaryotic yeast cells to recover and repair DNA damage in the absence of cosmic radiation. And that's important because one of the things that NASA is doing is sending what I call the ultraviolet astronaut into deep space, and that is a yeast cell. Because like I said before, they're ultimate astronaut because they're, if you think about it, they're a very bizarre organism. They're eukaryotic, so they're like us. They have a nucleus, they have mitochondria, they have all the stuff. They have same DNA repair systems. Right. The thing is, they don't need to eat. They don't need to have any water. Right. So they can be freeze-dried yeast are a living thing that don't need to eat. They don't need water. They don't care what temperature it is, and they're still alive. Wow. And they can sit there for weeks and months. And the idea is that every couple of weeks, some of these yeast cells that are on their own little spaceship are going to be woken up, and we're going to find out how much metabolism and DNA repair is going on as they accumulate vast amounts of cosmic radiation from deep outer space. We're doing the opposite experiment. We're going to see what happens when we take all that away. So it's very cool stuff. It's and
I mean, this would be in direct contradiction to LNT then, and it would. Totally. I've always been kind of skeptical of Hormesis. I think just because of my medical training is very LNT based. We were taught to be absolutely frightened about ordering CTs and X-rays that, over your career, you're going to kill this number of people through ordering medical diagnostic tests. So the idea of Hormesis just seemed kind of, particularly when I first heard about it, it sounded like a very fringe idea. There are studies that certainly seem to show a hormetic effect. I think the Cobalt 60 study in Taiwan, where they ended up building a building with a lot of contaminated rebar, and there was less cancer in those groups. But it just sounded sort of too far out, and too outside of my underlying dogmas. But I mean, some of the things you're describing in terms of these, ultra low dose radiation environments and that impact that you can actually see on a decrease in cell repair mechanisms is it's challenging my thinking on that.
Yeah. So with respect to that, I think I may have told you, we published a few papers a couple of years ago now, maybe 2 or 3 years back, where we tested the hypothesis that, if you are a cancer prone individual, in this case, mice, which are defective in a gene called P53, this is a protein that we call guardian of the genome. And P53 is very important for helping deal with damage caused by ionizing radiation. So we have a mouse strain that's very susceptible to getting cancer. It just gets cancer because of endogenous free radicals and natural background radiation. without having this gene intact. And what we did was we had the hypothesis that if all radiation is bad, even at low doses at the level of a CT scan, then clearly giving a single CT scan or multiple CT scans to mice that are already cancer prone is going to increase their risk. And the opposite effect was found. That's a hormetic effect. And we published those papers. And it's interesting because most hormetic effects are caused before an insult. So in other words, you could say, I want to adapt these cells by giving a hormetic treatment, and now they're going to become resistant to something bigger. And there's thousands of papers published on that shows that in fact works.
Yeah.
There's very few papers that say, okay, once you've been exposed to something that causes cancer, can you subsequently then reduce that risk by doing something after the fact?
Right.
So one of the experiments we did was we gave these mice a big dose of radiation, 4 gray, and we caused cancer in them for sure.
Yeah.
But then we waited about a month, I think it was a month or two, so 1 month. And then we started giving them CT scans, 1 CT scan a week for 10 weeks. Mm-hmm. And those mice that got that big cancer causing dose lived longer and had less cancer by getting more CT scans. So that's adding up dose.
Right.
There's more dose causing less risk. So you tell me what that means.
Yeah.
I think you'll figure it out. So, I mean, we're heading in that way in the future. Unfortunately, there's still a lot of doubt out there, and there's a lot of people that are worried about it for non-scientific reasons, which is understandable. People believe in all sorts of things, and that's, they're entitled to their opinions. And so I think the more we get a better handle on how this works, the more we're going to help people understand that maybe that they shouldn't be so concerned about things like getting a dental x-ray or a CT scan to feed a child or something.
So I wanted to maybe close with, this has been a really excellent broad ranging conversation, but something I did want to get to with you is I guess biases within the literature. I'm thinking about obesity research and how there was, such a dogma present there around, the effects of obesity on lifespan, for instance, and very much a kind of similar sort of linear modeling of mortality, say from obesity. And I think there's a consensus that actually, if you're slightly over that ideal body weight of, BMI, a body mass index of 25, that you actually live a little bit longer, if you're sort of 26 to 30. But it was kind of so, it was such a upset to the kind of dogma that exists that those kind of studies, even though they clearly show that in the conclusions, they'll actually say, no, the data fit the line well. And so I'm just wondering, I'm guessing hormesis, even amongst the radiobiology community, because of LNT, might be an idea that's fringe. So is there any sort of publication bias, or is there, rejection of papers because of that? Just to kind of question based again off that obesity research.
Yeah, well, probably the radiation literature is the classic example of that. I mean, there's confirmational bias everywhere, right? You can give the two scientists the same data, and based on their biases, they're going to interpret it one way or the other, right? Yeah. And so that's the problem that we have. The other problem we have is that it's a very confusing field to be in if you're not an expert in it because of the dosimetry and the doses and all the other confounding factors. That being said, it's the most highly studied thing that we've ever looked at in terms of human biology is ionizing radiation. It's been going on for years. So as the literature changes, there's definitely confirmational bias in there. There's going to be, I think that we've adopted the LNT for a practical purpose of radiation protection. It's easy for people to understand that keeping the dose as low as reasonably achievable is a prudent thing to do to keep people safe just in case. But when people start using that hypothesis or model to try and predict your risk as an individual from getting a CT scan, that's where it fails, and it should not be done. And a lot of people are now saying that, including some of the regulatory bodies, this LNT model cannot be used to predict risk. It can be used for radiation protection. Right.
And, I guess by exaggerating one risk over all others, that can lead to policy that is net harmful for society.
Correct.
And and that would definitely apply to, nuclear energy, which has big collective benefits in terms of clean air and its benefits for climate. But because of our exaggerated fears of radiation, that's led to, a regulatory ratchet that's made nuclear, very costly and not as cheap as coal as it used to be. Maybe just in closing, I kind of call it a choreography of fear, which is engaged with at nuclear waste sites or nuclear plants where you've got to put on all the booties, and there's this idea of reducing, because of that principle of the LNT, getting radiation exposures as low as reasonably achievable. I mean, we started this conversation off on the basis of kind of occupational health and your father's history. And it seems like there's been just such an overreaction. Like if you go to a coal mine, you're not putting on booties everywhere and wearing a respirator, despite the occupational risks being so much higher. But within the nuclear field, things are just kind of so exaggerated. And you gave that example of the lead apron when you're getting a dental X-ray. It's kind of created and really added to this fear that we all have, and that I have as a physician, that hey, I'm going to cause a cancer by ordering a certain number of CT scans in my life. I'm just wondering if you can comment on that, or if you have any kind of humorous anecdotes about that, again, choreography of fear.
Well, there's lots of examples of how that happens in society, right? I don't think we're going to be able to change everyone's opinion on the fear factor. I do believe that you're right. I mean, nuclear power is clean, it's safe. It's a way to absolutely, for the future, for climate change, we have to have more of it. And I think people are trying to advance it in a way that makes the average person less afraid of it, because of maybe these ideas of smaller things we can manage a little easier. It always comes down to the spent fuel, which some people call nuclear waste. I think a lot of people would argue that it's not waste, it's actually a commodity. There's lots of things that are valuable inside spent fuel. And so we have a lot of educating to do. I do know that some recent research that's been done has shown that people that work in the nuclear industry are actually more afraid of radiation in general than the average public, because the first thing that happens when you're in the nuclear industry is you start getting trained on how to avoid radiation, how it's going to be bad for you, how it's going to be this, why you need to take training on how to detect minute quantities of it, just to make sure you don't contaminate somebody or someone else. You walk into a nuclear power plant, like we had people walking into Chalk River or McMaster reactor here, setting off the whole body detectors, and they get all freaked out because they're radioactive, where it turns out to be just a radioactive tritium particle on them or something. And so we have the ability to detect this stuff at super low levels, like tiny amounts with high efficiency. And that causes people to be afraid, right? Because they can hear the Geiger counter going off. They can hear the whole body detector. Whereas all the other stuff that we can't detect in small amounts is all around us, and we don't really worry about it. Right. I think the classic way to end it is some of the stories you hear. The one story I heard about the injections for COVID-19 is, conspiracy theories. There's lots of them out there. And I was at the dental office talking about this dental X-ray thing, and the technician just seemed to say to me, well, the guy I just cleaned their teeth, and he's not getting the immunization because he's sure that there's Viagra in the injection. So what can you do about that kind of stuff, right? It's just a fear factor that people can create, and it's going to be impossible to change their opinion. Knowledge will help.
Yeah, no, that's a very interesting reflection, that people within the industry are more afraid of radiation perhaps than those without. I'd always kind of assume the opposite, that if you work in it, you'd understand it better. But I think absolutely that choreography of fear, and all of the detection and the ability to detect. Well,
that's just the stats, right? When you do sociological studies and you look at and you do survey people, the data shows that in fact the people that are told about it all the time and reminded about it all the time, and it's, I wouldn't say shoved down their throat, but basically, if you're pregnant, you got to get out of this nuclear power station because it's going to be dangerous to you, when that's not the case. Yeah. But we do these kind of things, and that instills fear. There are the opposite people that are anti-everything and anti-radiation. But then there's the general public. I would say, if you look at the statistics, I'm really worried about it. Health Canada has been trying for years to convince people that radon gas is going to be the second lead to lung cancer, but cause lung cancer in their homes. But then if you survey people, most people don't care. They're not really worried about radon because they're not told about it every day, right?
I have a friend who goes everywhere with a Geiger counter, and she left it on. She made the mistake of leaving it on at night, and like a cosmic ray just happened to collide with it and set off an alarm and woke her and her husband up. And her husband said, "God damn it, you're turning off the Geiger counter at night."
Just wear a tin, get some aluminum foil out of your cupboard and put it around your head. That protects your brain.
Yeah.
A lot of people do that.
Okay, Doug, a fascinating conversation. Pretty technical. I hope some of our listeners can follow along and understand some of the fascinating things we talked about today. Thanks again for making the time to come on Decouple.
Yeah, anytime, Chris. Thanks a lot.